Okay, good morning. I would like one of the GSIs to go into each of the side rooms to see if this is coming out okay. And if it isn't, can you come back and tell Dave in the booth about it? Okay, it's 10 after, so we're going to start. My name's Mike Moser. I'm the coordinator for Biology 1B, and I want to welcome you to the Biology 1B and wish you a Happy New Year. I think you're going to have an exciting time here. You have 3 really excellent instructors. Our sequence of the classes is going to be plants and fungi, and then evolution, and then ecology at the end. So my main job here is mostly administrative. My door is always open to you to come in. I have regular office hours, which are posted. The main thing I want to say is, because we have a lot to do today, is you're welcome to come and see me this morning or at any time, but you can save yourself a lot of trouble by going to our website, Biology 1B website, and to go onto the syllabus. That's the first thing I think I have up there. That will answer a lot of your questions. Now, during this week, I've gotten a lot of questions about how do I switch, how do I add, I'm on the wait list and all that. Hopefully the syllabus will deal with those. I've written up there the answer to a lot of those questions, I think. The bottom line is the only way you can switch or add is on telebears. We're shutting off telebears on Friday. Starting next week, there's no adding or switching. If you're on the wait list, don't come to class unless you're enrolled. And I stress that. Don't come in asking, well, I'll just hang around. We had some unfortunate situations where people were allowed to hang around in the middle of last semester. They found out they weren't enrolled and they'd wasted half a semester. So please, read the syllabus, read what's up here. If you have any questions, I'm in 2013. Get to know our webpage, and because that's where the information is. There's no lab manual. You'll be downloading all your stuff, and again, you'll be getting updates as time goes on. Your main contact in this will be the professors teaching and your GSI. Okay, my office is just around the corner. You're welcome to see me at any time, but you can save yourself probably a wait in a long line by trying to by reading this. Students repeating the class is lecture only. You can see me about that. Okay, so it's 12 after. I want to get going, so I want to introduce Professor Louis Feldman. He's going to be starting out for the first 5 weeks, 14 lectures, and have a great semester.
Good morning. As Mike said, my name is Louis Feldman. Before I begin the class, I want to introduce to you the third instructor who will be teaching the ecology section. He is Dr. George Roderick. George, would you stand up? George is in the environmental science policy and management department. He's a population biologist. He studies genetics, and he has done a lot of work in conservation biology. So many of you, perhaps some of you, will be interested in talking to him about a major. He, as we're both in the College of Natural Resources, and there are a lot of biological science majors, many more applied in the College of Natural Resources. So George will be here. He will be lecturing here the last third of the class, but I wanted you to see him. And a point I'd like to make about this class is, although Dr. Moritz, who is the second lecturer, isn't here, and although the class seems like it's divided into thirds, we try very hard to integrate the 3 portions of the class, to use examples that run through the entire class to help you see this as a coherent whole rather than as 3 separate units. So George, thanks for coming. You're welcome to stay, although you probably know it all already. I'd like to begin by giving you my email address, which is to my left on the board here. I will be glad to take your email questions. Sometimes the questions are really long and complicated. If they are, please make a time to see me, although I will try and answer them on email. I tend to get a flurry of emails just before the midterm, and so you might imagine if there are a lot of them, I might not get to your questions. So try and get me your emails earlier. My office hours are going to be in this building in room 2017 on Monday, Tuesdays, and Wednesdays. That is on Monday and Wednesdays right after lecture. They'll be from 9 to 10. And I'm glad to make an appointment, as I do often with students, if you cannot make those office hours. But by having them on those 3 days, we're able to get most of the students. And I'll talk a little bit more about office hours as we go on. Now, I want to tell you a little bit about my lecturing style to sort of help you. I've been up for about 3 or 4 hours. I've had a couple of cups of coffee. And students have asked, because I'm sort of excited about the subject, if we could have some warm-up exercises to get you up to speed. And the answer is we don't have enough time for that, unfortunately, because there's so much I want to convey to you about plants and fungi. So to slow me down and to make it possible for you to take notes, I write important points on the board. So the things that I write on the board are things that I expect for you to put into your notes or to have as information with regard to your notes. I will try very hard to speak slowly, and I will try hard to write clearly, but if something is illegible, raise your hand, and I'm really glad to address it. In addition to the notes that you're going to be taking here in lecture and from the board work, I will also have in your syllabus an outline of each of the lectures. The outlines are prepared before the class begins, so sometimes I modify them just before lecture, and they may not be exactly parallel with the lecture. But in most cases, they will be. So the outline will give you a direction of where you're going. And as well, in the syllabus, there are page numbers for the reading assignments that you might be interested in reading before the lecture. I should point out to you that the exam, as far as my questions, are going to be drawn solely from the lecture material. If there's something in the textbook that happens to be the same as the lecture, well, then it'll also be in the textbook. But the emphasis on the exams and the questions will be from the textbook, from the lecture, and not from the textbook. In addition, the third aid which we have for you is a webcast. Some of you have taken classes which are webcast before. This class, as you know, is at 8 o'clock in the morning, an hour that you find probably difficult to get up or to stomach. We have to have it at 8 o'clock because of the labs in order to accommodate all of the students from 9 to 12 and from 2 to 5. So if you miss a lecture, please use the webcast. The webcast is meant as a supplement for the lecture. But I want to remind you the difference between watching something on television and going to a live performance. At a live performance, your eyes can go wherever you want. On television or the webcast, you have to go where the person who's doing the monitoring directs the camera. And I should point out to you that sometimes the webcast doesn't work. It breaks, or the person is out of focus, or whatever. In that case, if you depend solely on the webcast, you're not going to be able to follow the lecture. So I want to encourage you to use the webcast, but use it as a supplement. And I'd like to encourage you to come to lecture, and I'll give you some strong reasons why. Now, this class is not graded on a curve. We will be happy to give everybody A's, and the grades will be 90 to 100 generally, or A, 80 to 90 is a B, and so forth. We have certain standards which we put out to you in the beginning of the class, and we have expectations based upon our years of experience with regard to your performance. So we believe that everybody could get A's if you apply yourself, and we're happy to give A's, but as you might imagine, it doesn't turn out that way. So remember, the only person you're competing against is yourself. And we want you to know that we're happy to accommodate you in whatever way we can. Now, to assist you in learning about plants and fungi, which probably for most of you represents a new topic, a topic probably that you haven't covered in your high school class or perhaps community college class, I bring in a lot of examples. And these are examples which are relevant to your real life. These are real life examples to help you make a connection between your daily activities and the biological talks that we have here in lecture. These are some examples that I brought up here to the front, and one of the reasons why I want you to use these examples is that learning is best accomplished by using as many of your senses as possible. You're sitting in the audience looking and seeing, but if you come up and you touch the organisms, or you touch the material, or you smell it, or in some cases you taste it, you're using many more of your senses, and that makes the concept that solidifies it much better in your mind. So I want to encourage you to take advantage of the fact that throughout the semester I'll be bringing in very unusual plants, and we'll be talking about them. And that brings up a point. I'm going to be telling you a lot of interesting stories. These are stories that I'm telling you for your enjoyment, for your pleasure, for your excitement. They are not meant to be tested on. So sometimes I'll say to you, put your pencils down and just listen to the story. It's not a test. It's mostly to sort of let you see the excitement and the wonder of nature. Now, in addition to the living material that I bring in, I'll also be showing you a lot of illustrations, slide material on Keynote or PowerPoint. Many of these pictures are taken from places here on campus, And the point behind it is that many of you have walked by these situations daily, and you'll continue to walk by these situations probably for the rest of the time you're at Berkeley. So that this is a chance to reinforce using real examples outside the material that we're going to be talking about in lecture. So I want you to take advantage of the fact that we're going to try very hard in many ways, many different ideas to help you appreciate and to conceptualize the material. I want to say that this is a class which is stressing understanding, conceptualization, not a class where there's memorization. There will be, unfortunately, some memorization of terms, but I'll try very hard to hold that down, and I think the other instructors will do the same. This is a class in which you have to take two ideas, perhaps, and put them together and come up with a third idea. It's a class where you demonstrate your thinking, not your memorization. And I know that's hard in some cases because you've been trained in high school to memorize. We want you now to understand and to appreciate. Okay, now you have a responsibility of being prepared for this class, and most particularly being prepared for the labs. The labs in Bio 1B and the lectures parallel each other. They reinforce each other. The lectures are an integral part of the course. They're not a separate class as they are in Bio 1A. So it's important that you prepare for the labs. That means you have to read the lab before the exercise. In some cases, in order to make this class more interactive, rather than me just talking to you, but you actually having an interaction, we ask you to collect some material for the lab exercises. You have to have read the lab then before you show up in the room, or you won't have collected the material. And your GSIs this year are going to take note of this, and there are going to be some questions with regard to the collection of the material. So make sure that you read the labs, that you come to the labs. Now, I come to all the lab sections during the portion of the class that I'm teaching. Many of you are going to feel embarrassed or intimidated when you see me walk into the room. My reason to come to the lab is to try and meet each of you, or as many of you as I can, in a smaller setting. This is a huge class. And in a smaller setting of the lab, I can talk to you. If you have questions, you can address them to me in this smaller setting. And I can also find out how well I'm doing or how poorly I'm doing with regard to conveying concepts. So what I want to say to you is if you see me walk into the lab, don't leave the room. Stay there. I'm really there to help you and not to embarrass you. Okay, with regard to the office hours, I want to say again that I am going to be available those days in the room, and a remarkably small percent of you take advantage of the office hours. Many of you feel you have to come in and ask some great question, but in fact you might just want to know why I decided to study plants, so you may have seen an interesting article in a newspaper which you'd like to discuss. Come into the room, get to know the instructor. I get to know about 50 students a year really well, and for many of them I can write a personal letter because I know these students well enough. So we provide the opportunities for you to meet us, and unfortunately, for whatever reason, many of you feel that you have to ask a very witty question in order to come to office hours. Sometimes you just want to come in and see what other questions are being asked by your peer students, just to see if you are thinking the same way they are. So these are sort of the outlines, the expectations of the class that we all have, and I want to make sure that you are on the same page with us. Are there any questions or ideas that you want to put forward before we start? Okay. So I'd first like to begin by putting out the questions, why should we study plants and fungi? Why in introductory biology should we take 1/6 of the material that we present you and focus in on plants and fungi? What are so unique and important about these organisms that we should focus on them for 1/6 of this class? I'd like to give you a couple of ideas. Some of you may have some other notions or answers. The first is that plants form the base of the food chain. All of the foods, all of the materials that we eat ultimately, except with very rare exceptions, originate via the process of photosynthesis, that is the capture of carbon dioxide using sunlight to make all of the organic compounds which we eat. This is an important process, and it makes relevant why it's important for you to understand and consider the plants that are outside. Secondly, the plants regenerate our oxygen atmosphere. This is a big problem these days with global warming. What's going to happen to the regeneration of the atmosphere with regard to oxygen and CO2? What's it going to do to plants when we increase the level of CO2 in the atmosphere? Thirdly, many of you are thinking of becoming practicing biologists, or you would like to. In order to be a well-trained and well-schooled biologist, you have to know something about the biota, the organisms which are out there. If you knew nothing about plants or fungi, you would be missing a great deal of the biota, a great deal of the understanding of the organisms. You can't be a conservation biologist, you can't be an ecologist unless you know something about the plants and the fungi. Some of you might be thinking of becoming physicians. I know there's a large percent in this audience. Many of the drugs which we use, our medicines, originate in plants. Many of you may come from East Asian or Asian cultures where plants are important in medicines. We're going to talk about a lot of those. Many of the diseases which humans get, especially fungal diseases, are a product of some of the organisms that we're going to consider today and that I will talk about. So these are some reasons that you might want to consider it. And last but not least, I want to say to you that there are going to be a number of social issues that you as voters are going to have to act on. For example, biofuels. Is this going to be a good use of land, to clear land to plant organisms and then convert to ethanol? And in a way, divert food crops because you're now not planting food on the land, you are actually planting fuel on the land. How about genetically modified foods? Do you want to eat genetically modified foods? Do you want your kids to eat genetically modified foods? You can't make the decision. You can't make an informed decision unless you understand something about the organisms which are involved. So these are some practical reasons which I'd like to put forward. There are many more. Some of you may have some your own ideas, and I'm glad to discuss them with you. Finally, I want to tell you something about why I've been so excited by plants, why I've studied plants for my entire career, why there's a whole department on this campus in which faculty study plants. So I'd like to convey for you some of the excitement. My responsibility is to present you with the passion that I have for these organisms and to give you a chance to go more deeply into them, to see something that you may not have known before, to become excited by this group and to convey to you some of my excitement. So during the first 5 weeks of this class, we're going to talk about plants and fungi. Now, the classification of organisms into the plants and the fungi began with a Swedish botanist by the name of Linnaeus. Linnaeus lived in the 18th century, and what he did for his taxonomy, he was a taxonomist, is he classified organisms initially as either animals or as plants. He said any organism which moved, which ate, which breathed, and which probably had a defined life cycle. That is, it went from juvenile to an adult. Organisms which had these characteristics in general, he would call an animal. And this is contrasted with organisms which there was no apparent movement, no eating, or nothing that could be viewed as traditional eating. There was no obvious breathing. And the life history was long, or in some cases, undefined. So organisms which had these characteristics, he then called plants. And based upon these two sort of large groupings, the first group of organisms that we're going to talk about, namely the fungi, were put with the plants. And today, as I will mention to you, because of this historical placement of the fungi with the plants, these organisms are still in the main on university campuses studied in plant biology departments. This is a historical grouping because of Linnaeus starting out. Now, this sort of classification scheme persisted up until the early 20th century, and by that point it was pretty obvious that it was more a sort of historical remnant. It wasn't really very relevant to what was going on in life. And at this time then, there was a sort of abandonment of the grouping of organisms solely as plants or animals. But again, the fungi are treated as plants or are discussed in the plant group because of historical reasons. I want to point this out to you. Now, we're going to talk today and also the beginning of next Friday on fungi. And what I would like to do is to introduce you to a few broad ideas or concepts which not only help us to understand and reinterpret the fungi, but also give you a basis for understanding all the other organisms in the biota and will help you both in this portion and in the two succeeding portions of the class. And these new notions which come about, which are important now in understanding and in treating the fungi, are the following. The first sort of general or overarching notion or theme that we should keep in mind is that we think the Earth formed between 4.2 to 4.5 billion years ago. The second notion is that we think that the first life evolved somewhere between 3.5 and 3.8 billion years ago. So at a minimum, only 400 million years might have needed to elapse for the first development of life. A really remarkably short period of time. We don't know if that's the case, but just based upon estimates which people have. And the third important or overarching development or conceptual theme which has influenced our understanding of organisms and which we will use to interpret and to characterize the fungi is that with the development of molecular techniques, we are gaining a better and an increased appreciation of the taxonomy of organisms. I'll put renewed because it may not necessarily be any better. So let's talk about now some of the new ideas or concepts which have come out of this new molecular approach to the evolution of organisms. And coming out of the molecular biology and being able to look at the DNA using molecular techniques is the current scheme of the grouping or the classification of organisms. And that scheme is the following. There is a common ancestor, and we could say maybe 3.8 billion years ago. And from this common ancestor, we have three great branches radiating. We have one branch which gives rise to the organisms which, for our purposes, we'll call the bacteria. Then we have another branch which started out from the common ancestor and then shortly after beginning branched again to yield two separate branches. This first branch is comprised of a group of organisms which are probably the least familiar to you. These are the archaea. And the last branch is the eukarya. Most, indeed, I would say all of the lectures that I'm going to be giving, and that will be followed by Dr. Moritz and Dr. Roderick, most of our attention will be focused on the eukarya branch of the scheme. And now what I'd like to do is to magnify the eukarya branch and to put some landmarks on the branch that we are going to be considering. So I'm going to put here the eukarya branch, the eukarya branch. At the bottom of the branch, we'd have the common ancestor. Common ancestor. And now let's put some landmarks on this branch, some things that you would recognize. The first thing that individuals always ask is, where are we? So near the top of the branch, we put the animals. The animals. And if we look at the first group of organisms that we were going to study, what we would find is that we would place the fungi at this part of the branch. A split sometime in the past, one branch led to the fungi and one branch led to the animals. Now originally, in Linnaeus's day, all of the organisms that he called fungi would have been included in this branch. But we now know, because of molecular techniques, that many of the organisms which were originally considered to be fungi are not fungi. They are different groups of organisms which appear different places on the eukarya branch. So I'd like to put some names of some groups. These are organisms which are formerly part of the true fungi and have been removed because they are shown to have very big differences in their DNA and therefore in their relation to these organisms. The first groups I just want to put on there that were removed, and we're going to be seeing examples of these in the lab, are the slime molds. And another group which is called the true slime molds, and you'll be able to see some differences if you take a look in your book. Another group which has been removed from the true fungi and which is important is grouped into a large called the stramenopiles. This is where you would find the organisms that bring about sudden oak death, the plant-killing disease of the oak trees here in the Bay Area and in much of California. So the point to make is that these organisms originally would have been put with the fungi, but have now been removed, and you can see placed on different portions of the eukarya branch. Now how about the plants, the other organisms which we are going to be studying? The plants would be placed here, and again, this grouping of plants was artificial. Linnaeus put into this plant grouping many things which we now know are not the true green plants. These are the organisms that we're going to call the plants. And so, for example, the red algae, which were initially grouped with the green plants, have now been removed because they are not part of the green plants with regard to their molecular sequences. Another group of organisms that were put with the plants, but we now know are not related to them, are the diatoms. So we're going to spend time going back and forth to this branch and looking at it carefully. Now there are a couple of points which I want to make about this branch. The first point, which I think is important for you to realize, is that animals and fungi, the true fungi, are fairly closely related. So fungi and animals are relatively closely related. That is, there's a common ancestor that they came from. This is an important point which we'll go back to very shortly. And the second point is one which I made before and which I want to emphasize again, is that many organisms which were originally grouped with the fungi and many organisms which were originally grouped with the green plants have now been removed, have been taken out of this group. And they are considered and treated because of historical reasons with the plants and with the fungi, but in fact they are not related with regard to their DNA or with regard to many of the other characteristics that make up their life history. So I want you to keep that in mind. This is an important point with regard to considering these organisms. Now fungi, we believe, have been around for about 500 million years. About 150,000 species have been identified and classified. And I'm treating fungi as a big group now, the old sort of Linnaeus sense. But in fact, it's been estimated that there are probably 1.5 million species of fungi out in the environment. So a large number which still need to be identified and which will be identified. This is a big task, and a lot of people are interested in studying the fungi. It's a very popular topic of study these days. Fungi come in all sizes and shapes, all different colors, and I'll show you some slides of these organisms very shortly. Some of you have already seen them. Now, when you, in a class like this, which is introductory, deal with a large group of organisms, to give you kind of a sense or an overall picture of these organisms, something you can carry away, something you can say, yes, these are the characteristics of fungi, what I have to do is give you generalities. Generalities are an unfortunate aspect of dealing with a large group in an introductory class, but also is going to present you with a way of making a picture of these organisms. So these generalities I give you apply to most of the organisms we're going to talk about most of the time, but there are some exceptions. So let's begin with some generalities with regard to the characteristics of fungi. And these are, again, generalities. The first grouping of generalities is the following. Fungi are non-motile, that is, no movement. They are generally, but not always, filamentous. That is, they make a filament. And you're going to have a chance to see this in the lab, where you have a series of cells connected to each other in a filament. Third, they are non-photosynthetic. That is, they don't use chlorophyll, they don't have chlorophyll to make any kind of carbon material. The second important basic characteristic of fungi is that they live in their food and absorb nutrients from dead or living matter. Now, how do they do this? Fungi do this by secreting enzymes, proteins. They secrete enzymes or proteins into the material in which they're growing, and these enzymes or proteins start to digest the organic matter, start to break it down, and then the fungus absorbs it. Fungi have in their cell wall a very unique compound. Some of you have heard of this before. The cell wall has in it a nitrogen-containing polysaccharide known as chitin. Anybody know where else you find chitin in the biota? Yes? Shells? How about another larger group? Insects. Very good. Okay, so you find them in insects. That's another place. So you might want to ask yourselves, how does this chitin come about in a very different group of organisms? The life cycle of fungi involves structures known as spores. Which are single cells capable of giving rise to a new organism. And the fifth characteristic, which I'm going to give you now and then I'll add to in a few minutes, is that their life cycle can be both sexual and asexual. Now, fungi, along with the heterotrophic bacteria, so fungi plus the heterotrophic bacteria plus a few other groups are the great decomposers of the environment. Their activities, that is, these decomposition activities, are as important for the life of this planet as the activities of plants which synthesize carbon compounds. Because through the decomposition, we release CO2 back into the atmosphere, which may be a bad thing these days, and also nutrients and minerals back into the soil. So through these enzymes which the fungi produce, they involve or they start decomposition. It's a very important part of the life cycle here on Earth. Now, fungus growing in a forest with these enzymes that cause decomposition makes no distinction between a tree which has fallen and is beginning to rot and a fence post which you may have put up in the forest. Equipped with a very powerful arsenal of digestive enzymes, we believe that fungi potentially can digest any naturally organic compounds which are in the environment. And in fact, many of the synthesized organic compounds we now know fungi can digest. So fungi can be extremely destructive. Some of you may have personal experience with damage due to fungi, dry rot, for example, in a house. These are fungal degradation activities. But the qualities, namely the enzymes that are secreted by fungi, which make them such pests, also are important in their use, positive use by humans. The enzymes which are secreted not only change many activities in the organism which is either being eaten by the fungus, but also release compounds which are important. The basis of the baking industry, for example, is a result of CO2 being produced as a result of the yeast, a unicellular fungus, digesting the sugar and releasing CO2. The basis of the brewing industry is a result of ethanol being produced because the fungus has been digesting the complicated carbohydrates, converting it into compounds which eventually turn to ethanol. So humans have made for great use of fungi and their activities for thousands of years. The basis of the cheese industry. Before refrigeration, which was a relatively new phenomenon, was brought to the market, most dairy products would spoil until it was discovered that if you take the dairy product and you inoculate it, or by chance a fungus grows in it, with a fungus, that the fungus actually will help to preserve the products. So, for example, this is blue cheese here. If you come up and look at blue cheese, a particular type of fungus has been inoculated into the milk and has converted the cheese into a product now which is more stable and less likely to be attacked by bacteria. The green color that you see, or the blue color you see in the cheese, the veins as you sometimes call them, represent masses of spores that this particular fungus is producing. The fungus also flavors the cheese. Often acids of various types are produced, such as malic acid or citric acid, and other compounds which give cheese their distinctive flavor. Camembert cheese, if any of you have ever been to a factory where they make camembert cheese, there is a smell of ammonia in the air. Ammonia is probably a byproduct of this fungus and is probably used by this fungus to retard bacteria or other organisms from growing on the fungus. So fungi, a lot of the lore about fungi have to do with their use in food. As well, though, a lot of the lore about fungi have to do with the fact that they produce dangerous toxins. And many of you have heard of the fungus known as Amanita. Amanita, which is sometimes known as the death angel. Every year in the Bay Area here, there are reports of people who go out in the hills and collect Amanita and go home and cook it and then die. The reason they die is because this fungus produces a toxin which is not denatured by heating and which causes irreparable liver damage. And the only way these individuals can be saved is with a liver transplant. And if it rains this year, we'll probably see in the newspaper, especially the Chronicle, big headlines of people who die from eating these mushrooms. Fungi also produce a large number of compounds which are toxic for us. And we have to assume that these compounds have some evolutionary advantage, perhaps preventing some bad organism from consuming the fungus. And one compound which is of particular interest to us is known as aflatoxin. Is a fungal toxin which is produced. Some of you may have an allergy to aflatoxin. This is a toxin produced by fungi which, among other organisms, grows on peanuts. And peanuts are allowed to have a certain level of contamination by the U.S. Department of Agriculture of aflatoxin, a very low level, which for most people does not cause problems. But for an individual who has had hepatitis B or C, consuming any aflatoxin increases their risk greatly of cancer. So many individuals have to avoid even touching peanuts, have to avoid even using any dishes which have had peanuts in it, because the low levels of aflatoxin can prove particularly dangerous for them. Many of the products which are produced by fungi, which are considered perhaps bad, are, as it turns out, good. Here is a petri dish, if some of you have looked at it or want to come up look at it, which contains the fungus penicillin, or penicillium, which makes the antibiotic penicillin. This is a compound that the fungus probably produces to retard bacteria from growing in its food or near it. And we as humans, unlike the bacteria, are in general not allergic to the penicillin, and thereby can use this as an antibacterial agent in our lives. Another compound produced by fungi, which is probably important in their life cycle, is cyclosporin. This is a fungal toxin, but it's a fungal toxin which humans have used in transplants. It prevents rejection. So many of the compounds that are produced by fungi are exceedingly useful for our work. Now, fungi can cause a lot of diseases in humans. Ringworm, diaper rash, toenail fungus. If any of you are from the Central Valley of California, you're familiar with San Joaquin Valley fever. These are all fungal diseases which are caused by fungi growing in our bodies. In most cases, we are able to, with a little help, our immune system is able to fight off the fungal diseases. But in some cases, and especially in the past before drugs were developed for HIV/AIDS, most of the people who had HIV/AIDS whose immune systems were compromised died of fungal pneumonia. And they died of pneumonia induced by fungi because their immune systems were unable to fight off these particular organisms. So this is an important group of organisms. For those of you who are thinking of becoming physicians, it is important to be able to understand something about their diseases. Now, how do we make drugs to counteract fungal diseases? Well, part of the problem which we have in making a drug to counteract fungal diseases is that fungi and animals are very closely related. We share, believe it or not, a lot of our genes, a lot of our biochemical pathways. And so in the development of drugs, we want to develop a drug which targets the fungus, but which doesn't also poison us. And in this particular situation, in order to develop a drug which kills the fungus but doesn't kill us, first of all, we have to know which fungus is growing. And then secondly, we have to know something about its biochemistry. How does it differ from us in order for us to design drugs to kill that fungus? And to give you an example, I just want to give you one. There are other examples, but I should tell you it's exceedingly difficult to get rid of fungal diseases. If any of you have a toenail fungus, for which I'll show you a picture later on, you'll know it's hard to get rid of this fungal disease because the drugs have to not hurt us. And even if you take a fungal drug, people still have to have their liver function checked because that fungal drug still has some effect on us. One of the drugs which has been developed to affect a pathway which results in the production of compounds that are used in fungal membranes. In our membranes, in animal membranes, we use cholesterol in our membranes. In fungi, in their membranes, they don't have cholesterol. They have a compound known as agosterol. And so by targeting a pathway which leads to agosterol, people have been able to develop drugs which have been used with regard to the fungi. Now, if we are lucky, I'd like to show you a few pictures in the last 5 minutes of the class to talk to you about the fungi. Okay, the first slide, I want to just show you some examples of the different types of fungi which we see. These are fungi which you can find growing around the area here, and if we get some rain this year, you will be able to view some of these fungi. This is known as it's not very big, it's a couple inches across, but it grows on rotting wood, and this fungus is then secreting enzymes into the wood and digesting it. This is a fungus which is fairly common, which grows on dead or dying wood stumps, and again absorbs the material from the wood by secreting enzymes and by breaking it down. Another fungus which is differently shaped, which seems to be growing from the ground, but in fact is growing from some organic material, is this one. Here's one which is known because it's like a puffball, and what happens is in the central portion, raindrops hit this sort of rounded white dome on the outside and cause the spores to shoot up. Now, here is a fungus that many people use for food. This is another fungus which is highly prized, morels, which is used for food. And this is the death angel, the one Amanita, which we talked about before. Here is the fungus which causes ringworm, a human disease. You can see it's a filamentous fungus. And here is a ringworm. I took out the pictures that look really gross, and just to give you an example of what the ringworm looks like if you have it on your skin. Here is toenail fungus, one which is very hard to get rid of. And here is the fungus which actually is involved in degrading your doors and your wood that you have at home. Here is a eucalyptus tree from outside, or was a eucalyptus tree from outside behind VLSB, which was blown down in the wind, and it was attacked by fungi. And I want the arrow points to a lamp, which was knocked off, and probably on days where it is very windy or stormy, probably not a good idea to walk through the eucalyptus grove. And then finally, these last two pictures show you some other diseases which are important. This disease is known as a rust disease, and this particular rust disease is a very significant disease for plants. And then finally, the last slide I want to show you is a disease which inhabits corn kernels. And if any of you are from Mexican or South American cultures, although we in North America usually would throw this away, in cultures from Central America and South America, this corn, which has this fungus that has taken over the kernel and now fills the kernel with spores, is actually roasted and is consumed and is considered to be a delicacy. Okay, we'll see you then all on Friday.